Welcome to the third day of the 17th Vermont Organics Recycling Summit, organized by the Composting Association of Vermont in partnership with Vermont Agency of Natural Resources. I'm Natasha Duarte, the director of the Composting Association of Vermont. And this year's summit is organized as a kickoff event of sorts for International Compost Awareness Week, which starts on Sunday, the 7th, and is the largest and most comprehensive education initiative of the compost industry. The theme for this year is for healthier soil, healthier food, compost. And I just want to give a shout out to Dr. Deb Nair's Composting Ecology and Management class students who do this sort of International Compost Awareness Week poster project every year. And the full collection, that's this poster right here on the right. And the full collection of these posters are available on the CAV website at compostingvermont.org/VORS. And if you click on the images on the website, you'll get to read the stories that the students wrote about their inspiration for the different posters that they made. So that's a cool project that we always like to highlight and promote. I'd also like to thank all of our wonderful sponsors who allow us to bring this full week program to participants free of charge, which we found has really increased accessibility and participation from a much wider community, which is great to get as many folks interested and into these topics as possible. I think that's part of the culture change solution for promoting successful organic management. Our sponsors this year are Community Bank, Nature Cycle, Eco Products, Vegware, Addison County Solid Waste Management District, AgriLab Technologies, Vermont Natural Ag Products, the Vermont Produce Program of Vermont Agency of Agriculture, Food and Markets, and RMI. So huge thanks to them, as always, for helping us put on this great program. We are in the 1 o'clock session, of course, today on May 3rd, Microplastics in Composts, Digestates and Food Waste, Findings from Vermont and Beyond. I'm going to stop sharing my screen here, and I'm going to introduce our presenters as Sarah brings her screen up. So we're joined today by Sarah Hobson, who is a master's student in the Environmental Microbiome Engineering Research Group and Department of Civil and Environmental Engineering at the University of Vermont. Her current research focuses on the quantification of microplastics in composts across Vermont. She's an environmental engineer in training and a lifelong composter with an international background. Sarah is engaged in local food recovery and mutual aid-based community efforts. Her co-presenter is Kate Porterfield, who is a PhD student in the Nutrient Cycling and Ecological Design Lab and Department of Civil and Environmental Engineering, also at the University of Vermont. Her research focuses on the use of ecological engineering to address environmental issues at the water, food, energy nexus. Kate has worked with an interdisciplinary team of professors and private industry members to evaluate the potential to recover and reuse phosphorus from anaerobically digested dairy manure. More recently, she has pioneered measurement of microplastics in digestate and food waste in Vermont, while leading a literature review on microplastic contamination and organic recycling. So thank you both so much for joining us today and being part of the Summit's program, and I'll turn it over to you.
Thank you so much for that introduction, Natasha. Kate and I are really excited to be here presenting about our work. And specifically, we're going to be talking about our measurements of microplastics and plastics in composts, both compost and digestates across Vermont. And we'll also be relating this to current literature and findings as well. And this was in collaboration with Dr. Matthew Scarborough, Deb Nair, Meredith Niles and Eric Roy, and this was done at the University of Vermont. So today's presentation will, see if our slides are going to advance for us. We'll cover kind of a background. So many of you are well acquainted with the organics recycling context of Vermont. We'll just briefly touch on that before getting into our research questions. I'm sure a lot of you have thought about plastic in compost and digestate. So we were thinking about that too, and we're going to going to provide some more specifics on the work we did and the methods we used to conduct our experimentation. And then we're going to kind of present our results and take a step back and contextualize it to current findings and what our results might mean moving forward.
Great. Thanks for that introduction, Sarah. I just wanted to say briefly, I see a lot of familiar names on this list, which is really exciting. I know many of you have heard Sarah and I speak about our research in the past. So if it's a little bit of repetition for you all, we apologize. But we're excited to have this longer block of time so that we can hopefully get into some more interesting questions and discussion at the end. So we're going to present for about half an hour and then we'll open it up to questions. So just going to start with a little bit of overarching context for everyone, starting with what's precipitated this increased interest in looking at microplastics in compost. I think part of it is the increase in organics diversion bans across the US and abroad. So more and more people are becoming aware, that when we put food waste in a landfill, we're not only emitting methane into the atmosphere, but we're also losing the opportunity to recover valuable organic matter and nutrients that's embodied in that food to reuse in the agricultural system. So I think this knowledge has prompted a lot of these new organics diversion mandates that are cropping up all over the place. So this nice map by the US Composting Council shows different states that have some sort of organics diversion initiative as of 2021. So different types of initiatives online right now. Some of them only apply to green waste. Those are the green ones. And then others are more all-encompassing in terms of considering other types of organic waste, mainly food waste and food scrap. So those are the orange and blue ones. The state of Vermont, where we're housed, has, I think, maybe the most ambitious organics diversion mandate in the United States, in that it also applies to household food waste. So in the state of Vermont, all food waste producers, even folks at the household level, are not supposed to have their food scraps go to the landfill. And maybe we can go to the next slide now. Yeah, so this is the Universal Recycling Law, which I'm sure a lot of people on here are really familiar with. And it mandates the diversion of all organics from landfills by 2020. And in Vermont, we have our own version of the food recovery hierarchy that's adapted from the US EPA's. And it goes from source reduction down to energy recovery in order of preference. In practice, a lot of food waste that's not suitable for human consumption or for animal consumption is now being diverted to composting or anaerobic digestion. And we won't go too much into what composting and anaerobic digestion are, because I'm sure you're all really familiar with it. But basically, composting, we're breaking down organic materials in the presence of oxygen. Anaerobic digestion, we're breaking down organic materials in the absence of oxygen. So one issue that's emerged as more and more folks have, been trying to divert their food waste away from landfills into organics recycling strategies is the possibility of increasing plastic contamination in compost and digestates. And that's because a lot of food waste streams are mixed with plastic packaging for one reason or the other. So we can go to the next slide. So this can be pre-consumer food waste streams, so food waste that has never even reached the consumer yet. We have a picture on the slide here of a local food manufacturer here in Vermont. This is just one example of the type of food waste that comes very mixed with plastic packaging. So sometimes, for whatever reason, food manufacturers have a run that is off specification and it's not fit for human consumption. And so then, in order to recycle those organics, we need to get the plastic packaging apart from the organic materials. And then there's another kind of food waste stream that can be mixed with organic packaging, but more unintentionally. So post-consumer source-separated organics are supposed to not contain any plastic packaging, but that is often not the case in practice, people are imperfect, and we can end up with PLU stickers or plastic bags or containers or any sort of plastic packaging in compost and organics collection buckets. And then in this picture, as you can see, it's also increasingly common to have compostable plastics mixed in with food waste streams, either intentionally or unintentionally. Next slide. So one strategy that's arisen to try and address some of these types of mixed food waste streams is the use of mechanical depackagers. So there are many types of mechanical depackager on the market right now. They are not super common in the US at the moment, but they are expected to become increasingly common as more states mandate organics diversion and practitioners are forced to deal with larger and larger volumes of packaged food waste. And so basically what these machines do is you feed packaged food waste into a hopper, and then it goes into a barrel, and some combination of low force paddles and screens is used to separate the organics from the residual packaging. So the use of this type of technology has been a source of a lot of debate in the state of Vermont over the past couple of years. And we won't get too much into that during this discussion, but I just wanted to provide that context because one of the types of organic waste that we measured for our study was mechanically depackaged food waste. So when we're talking about that, we're talking about food waste that has been passed through a mechanical depackager, such as this one. And the manufacturers of these mechanical depackagers report organics recovery and purity rates greater than 99% by weight. So they get most of the plastic packaging out, but it's possible that some passes through.
So depackagers are a big consideration when looking at digestion and digestate, but in Vermont, we should also make other considerations for compost, as depackagers aren't as common for compost operations here. So specifically, we should kind of look at the feedstocks that we're considering for compost. And there are quite a variety of feedstocks, as you all know, and here are some of the common ones, including your higher carbon bulking agents. But just to note that it's not just food waste that could be potentially introducing plastic. Any other feedstock might also convey plastic to the finished product. This could be, for example, shreds from construction waste. There might be some plastic left from hay baling that could be mixed in with hay or straw, or it could be in the animal manure if an animal had consumed any amount of plastic. So this is just kind of to mention that food waste might not be the only source of plastic. And then compost might not be the only source of plastic going to soil if it is one. In this presentation, we're kind of focusing on the potential for organic amendments, including compost and digestate, to transmit plastic to the soil. But there are also, for example, agricultural plastics like plastic mulching. Maybe driving the countryside, we're seeing increasing amounts of plastic just covering the soil, and this provides a variety of benefits, but we should also consider that these plastics might fragment and contaminate our soils over time. There's also plastic and food waste, which we touched on, and this kind of introduces the cyclical nature of plastic in our food system. So food might be packaged in plastic and then thrown away with that packaging, converted into compost that might have some plastic that is then added to our soils to grow more food that's, again, packaged in plastic. But outside of the food system, we also might have plastic in our waterways. A common example of this is fibers that are shed from our clothes when we do laundry, when we launder plastic clothes. Litter, this is kind of a very direct primary source of plastic contamination in our soils. Other equipment and also vehicles, tire wear contributes to plastic. Plastic is also kind of in the air that might be depositing all over, especially in our cities. A lot of dust tends to contribute to plastic deposition, and it might also be in our biosolids. So from these wastewater treatment facilities, and these are often spread onto our agricultural soils as well. So when this plastic enters our soil, it can have a variety of impacts. It can have some physical impacts. So it might change the soil water dynamics. Plastic is pretty hydrophobic and it repels water. It also might create different kind of pores and pockets in the soil that change the air dynamics of the soil. Soil aggregation is a pretty important one, and studies have found that plastic particles might reduce aggregate stability and size. So there are a variety of physical implications of plastic in our soil, and most of the studies that have looked at this look at really high concentrations of plastic, which we're not really seeing currently, but might in the future. And then in turn, this might have chemical implications for our soils. So changes in cycling of key nutrients like carbon and phosphorus and nitrogen, those might change with changing air and water dynamics, for example. And then the way that these nutrients cycle in the soils might change their transportation or absorption to different particles. And then these can also have biological implications. So studies have documented changes in microbial communities, species richness and diversity, the overall functioning of these microbial communities in terms of, for example, chemical cycling. And just to note that these are kind of all intertwined. So plastic, while it's not a traditional toxicant, does or may have a variety of impacts on our ecosystems. And this is kind of a developing field. We're still kind of understanding what the real implications of plastic might be with current concentrations of plastics in our soils and potential future concentrations as loadings increase and plastics accumulate in our soils. And just to note that there's also a time factor to this. So over time, plastics break into smaller and smaller pieces through a variety of mechanisms. There are 4 main ones. Photodegradation, so those UV rays that comes with weathering of plastics in the environment. Hydrolytic degradation, so this is kind of the splicing or breaking apart of those carbon backbones of plastic particles. Biological degradation, so these are those microbes coming to play and breaking down or breaking apart the plastic particles into carbon intermediates, and then thermooxidative degradation, and this is kind of more of a chemical process with radicalization of particles. And this just creates smaller and more numerous particles. And these are called microplastics. So you've probably heard of this term before, but just to kind of acquaint everyone, plastics that we call microplastics are in the size range of 5 millimeters down to 0.01 millimeters. So just for reference, we can reliably see with our eyes down to 1 millimeter. So when we're talking about a tenth or 100th of a millimeter, that's quite small, and we can't really see that with our eye reliably. And we rely on instrumentation for those measurements. But microplastics are just one of a few common size designations for plastic. There are also macroplastics, which are your really big ones, meso, which are kind of more medium-sized, and nanoplastics, those are the smallest ones that are commonly studied. But we're still kind of developing methods because they're quite difficult to handle and quantify. And there are also some common categories as far as the shape of microplastics. So these are fibers, which you can see in the images to the right. That's image A and D. And these are from our study. Fibers are kind of long in one dimension only. And these are kind of, they might be from our clothing. They're often quite numerous because they're small, but they're quite low in mass. There are also fragments. These are kind of your bulkier, chunkier particles, might be higher in mass. You can see an example in image B of a quite a large fragment that we found. And then as far as films, these are kind of thinking of plastic bags, maybe pretty long and planar, but thin. These might contribute a pretty low mass as well, and might be common in food packaging. And then as far as plastic types, the other consideration for plastic quantification, there are two major categories that we split plastics into: thermoplastics, which are your common recyclable plastics that can be remolded. And you might be familiar with polyethylene, polypropylene, maybe some PVC, polystyrene. These are common in food packaging, especially. And then thermosets, these ones, they're molded once and they're kind of set. There are ways to recycle them, but they're not used as commonly. And you might be familiar with epoxy or polyester, but these are kind of more niche engineering plastics that we didn't come across as frequently. And just to take a step back for the impetus of this study, there's been a lot of concern for microplastics across media. It's kind of become a buzzword across younger generations, especially concern for plastics in our food systems, potentially entering our bodies. So there's quite a lot of interest in microplastics in general. And Kate's going to talk a bit about the work that we've done.
Great, thanks, Sarah. So Sarah and I have been working on microplastics in one way or another for about 2 years now. Our work has taken two main tracks. The first was to figure out what other folks have done already and kind of what's the state of knowledge on microplastics in organic residuals from around the world. And so to answer this question, we conducted a literature review, and we compiled that literature review into a paper that we've put out pretty recently. And so throughout this presentation, we're going to use the results from that literature review to kind of contextualize our own findings within the broader body of literature. And then the second avenue of investigation has been to collect our own primary data. So we've measured plastic content in organic residuals in Vermont, including depackaged, so mechanically depackaged, source-separated food waste, mechanically depackaged pre-consumer food waste, and for that we used ice cream pints as our example, digestate that has been derived in part from those depackaged ice cream. So what's coming out of the back end of an anaerobic digester that is accepting depackaged ice cream pints. And then two categories of compost. So compost that is derived mostly from green waste, so manure, yard clippings, basically anything that's not food waste. And then compost that has been derived in part from some amount of source-separated food waste. And so to measure the microplastics in these materials, we first had to develop some sort of method to do that, because there are not really standard methods out there for measuring microplastics or plastics in these materials. And so we looked at the papers from our literature review that kind of, measured plastics in the most similar materials and used methods that were pretty cheap and straightforward and easy for us to replicate. And so I'll briefly go through the different phases of that plastic measurement method now. So the first one is obviously to collect the samples. So we've collected samples of organic materials from along the food waste management spectrum. So compost, depackaged food waste, and then digestate. And then once we have those samples, we bring them back to the lab. And we take smaller subsamples from them, and we try to reduce the organic matter so that we can get at the plastics that are contained in that sample. And so the way we do that is by taking a smaller subsample, we dry it in the oven, and then we add really concentrated hydrogen peroxide, which reacts with the organic matter in the sample. and reduces some of it, but also bleaches a lot of it. So it can make it easier to see the plastics against the organic material background. And so once we've let that hydrogen peroxide react with our sample for a couple days, we pour that mixture through these custom sieves that we make that you can see on the bottom of the screen. And the sieves we stack in 2 mesh sizes, so 0.5, half a millimeter, and then a millimeter. And so this tells us 2 things. Our lower detection limit for this study is half a millimeter. So we were not able to quantify microplastics below that size. There are definitely microplastics below that size, but we did not measure them for this study. We measured everything above half a millimeter. And then sieving like this also accomplishes dividing our plastics up into different size fractions. So we broke them into 3 categories: half a millimeter to a millimeter, 1 to 5 millimeters, and then above 5 millimeters. And everything that's above 5 millimeters is technically a macroplastic or a mesoplastic, depending on who you ask. But we measured those too, because those are also plastics that are in the material. So we thought that was important to quantify as well. So when we talk about our plastic, our results, we're talking about the total plastic content that we measured in these materials, including everything from half a millimeter and above. And so the last step is to actually identify the plastic particles on these sieves once we've gone through all those prior steps. And to do that, we put each sieve on a light microscope and enhance it to 40 times magnification. And then we go through with forceps and we pull out everything that could be a microplastic using kind of some common heuristics for identifying plastic from organic materials. And then we take those potential microplastics, and for a subset or all of them, depending on how many there are, we do something called ATR-FTIR, which stands for like attenuated total reflectance Fourier transform infrared spectroscopy, which is a really long name. But basically all it is this instrument allows us to take a particle and we put it on there and we shoot a beam of light through it. And then that gives us back a wavelength, and we can take that wavelength and then see if it has any matches in a library of known wavelengths of known materials. So this can tell us, is this particle actually a plastic? And if so, what type of plastic is it? Is it polypropylene? Is it polyethylene, polystyrene? And so we did that. And then we had one final step. So all of those steps get us up to an estimate of a number of particles per dry unit of material. And the reason that we normalize everything to the dry unit of material is because, as we know, you can have really different moisture contents in different organic materials. So we're always presenting the plastic content in terms of dry content of material to make it a more fair comparison. But so we get to our final number, and it will be, for example, 1,000 particles per kilogram of dry material. But we were also interested in knowing, what does that mean in terms of like the percent weight of the material? What percent of that material is made up of plastic? And getting at that is a little bit harder than you might expect. A lot of the time, these particles that we're pulling out are really small and they're hard to manipulate, even with our really fine-toothed forceps. And some of them won't even register on a microbalance. So when we would measure the particles directly weigh them. And then when we couldn't do that, we use assumptions about the dimensions of the particle, the size, the shape, the polymer type, and we estimate the mass of that particle. And so using a combination of those 2 methods, we converted our counts of plastics in each material to an estimate of the weight plastic composition of that material.
Okay, thanks for walking us through those methods, Kate. I'm going to be talking about some of the results, especially the results for compost. So here are some examples of particles that we found in the compost and digestate. This is an expanded polystyrene fragment. This is PET. PLA, polylactic acid. This is a compostable plastic. And this one was found in digestate. We didn't really find compostable plastics in the compost with confidence. This is a PE film. It was quite a large film, but there were quite a few other types of films we found, and this is just some examples. These aren't quite representative of all of the particles we found, but you might see that with this magnification, these two films I've just shown have the kind of a print pattern that's a pretty anthropomorphic sign, so kind of easier to detect than other fragments that might be clear or a color that kind of blends in with the organic matrix a bit more. PBAT, another common compostable plastic or decomposable, and then another film with that same print on it. And of these, we found films and fibers to have the highest abundance across digestate and compost, but the fragments and also larger films contributed the most towards mass. And I'll get into that a bit more in a few later slides. But to kind of put the quantities into more of a visual representation, here's a graph of our compost counts of plastic. You can see on the y-axis, that's the count of plastics per dry kilogram of compost. And on the x-axis, it's the 20 different compost samples that we measured from 20 unique composts. And the range was kind of from 100 to 300 particles of plastic. And this is for all sizes. So 0.5 millimeter up to larger than 5 millimeters per dry kilogram of compost. And this is pretty typical across the literature. And I just want to note that the highest values we found are just an order of magnitude larger than kind of the smallest typical counts that we found. And then when we looked at mass to contextualize the count, we found quite low masses. So kind of on the y-axis, we have the percent weight of plastic per weight of compost, again, on a dry basis for the 20 compost samples, and found plastic weights on a percentage basis from 0.0005 up to 0.00015. So these are quite small. But just to note that even the compost with the largest amounts of plastic on a per weight basis had, 0.1% of plastic or less. So another thing we really looked at was the shape. So you might remember from earlier, there are these three common shapes that we categorize plastic into: fibers, films, and fragments. And here I've kind of shown the proportion of shape for that total count or total mass, count on the left, mass on the right, for the three different size ranges that we looked at. So 0.5 millimeter to 1 millimeter, 1 to 5 millimeters, and 5 millimeters and above. And you can see here that while fibers were quite high in count, especially for the smaller size ranges, when we looked at mass, it's quite a small proportion of the total mass, and that's just because fibers are often quite lightweight, whereas fragments are usually the heaviest shape of plastic. And while we didn't count very many, there was an outsized representation of fragments when we look at the mass. So this kind of hints that it's important to just keep in mind both count and mass when we're quantifying plastics. And then when it comes to plastic types, we found that PVC, which has the highest density of the plastics we found, it had a kind of an outsized representation again for the mass because of that high density. But then surprisingly, polystyrene, which has a pretty low density, also had an outsized representation for the mass compared to the count. And that's because of the shape. So polystyrene was most commonly found as fragments, which are a heavier shape than, say, fibers. Whereas polyamide, for example, which kind of has a medium density compared to the other plastics. So you would expect it to have like a medium impact on the mass, was actually quite low in the total mass. And that's because most of the particles found for polyamide were fibers, which are quite low in mass, while very numerous in count. And now Kate's gonna kind of walk us through kind of more numeric comparisons of our findings and relate them to the literature.
Yeah, so we're just going to build up a table that's going to help us compare Sarah's findings of plastic content of compost to what others have found in the literature, and then to other types of organic residuals like digestate and food waste. So to start, we're going to give you an overview of what we found in the literature in terms of the plastic content of compost. So when we look at green waste derived compost, other folks have found plastic contents ranging from a tiny fraction of a percentage up to 1%. When we look at compost that's derived in part from source separated organic waste, so including food waste, we find like kind of a similar range. So ranging from a small fraction of a percent up to another small fraction of a percent. And then if we compare this to what Sarah found for compost in the state of Vermont, we see that Sarah's range of values kind of falls within what others in the literature have reported. So Sarah's values range from no plastic being detected to a small fraction of a percent of the material being composed of plastic. And then same thing for compost that's derived in part from source separated organic waste. And I will just point out here that though the lower bounds are the same for both of these categories, we did find about 5 times more plastic in the compost derived from source separated organic waste in terms of the upper bound. So the high was higher when food waste was present in compost in our studies. And then when we look at digestate, we see kind of a similar range of plastic content in the literature. So 0.01% up to about 0.25%. And then if we compare that to what we found for digestate in Vermont, we can see that our range falls roughly within what others have reported. And then if we look at food waste alone, or at the source of one of the potential sources of plastic in these materials, we see kind of a lower bound. So some folks have detected very low plastic content in source separated organic residuals. But in at least one instance, there is 6% of the material the source separated material was composed of plastic. So this makes sense to me that you can see really high variability in the plastic content of source separated organic waste, depending on a lot of factors like, how hands-on the program is, whether or not the collection is going on in a residential setting where there's a lot of buy-in, or if it's an institutional setting where there's more anonymity, and people are less careful about what they're putting in what bin. And then there was actually one other study that looked at mechanically depackaged source separated organic waste, and they found that the plastic content in that ranged from small fractions of a percent. And then if you compare that to what we found in depackaged source separated organic waste, we found very similar ranges of plastic in that material to what was found in the other study, so 0.014 to 0.12%. And then in the mechanically depackaged ice cream pints, we found slightly more plastic on average. So the ranges there was 0.066 to 0.35%. All right, so we just threw a lot of numbers at you, but Sarah and I wanted to point out We're trying to make these comparisons because I think everyone wants to know, like, how do these different materials compare? But we just wanted to point out a couple considerations when you're making direct comparisons in terms of plastic content of different organic residuals. One is that depending on the organic management strategy, we can have variable organic matter reduction. So if you have a strategy that is reducing a lot of the organic matter content, that could tend to concentrate the microplastics in that material. So it could look like that material has a really high plastic content if you compare it to one where there was less organic matter reduction, even if the starting amount of plastic in the two materials was the same. The second point we wanted to make was that this can also happen in terms of bulking agent dilution. So in composting, we're often adding some sort of a carbon source that might be from a relatively plastic-free, something like wood chips or sawdust or something like that. And depending on how much of that relatively clean material you're adding to your potentially more contaminated material, you could dilute it more or less. And the same thing goes for digestate. If a digester's mainly managing like pretty clean brewery waste, and then just a small, percentage of their feedstock is some sort of food waste, then that would tend to dilute the plastic concentration too. So we just wanted to add those two caveats to making direct comparisons. There's kind of a lot of factors that go into what plastic content we measure in these materials, and making direct comparisons doesn't necessarily help us understand what practice is ultimately going to have the least flow of microplastics to agricultural soils. For that, we need to take more of a mass balance approach. Okay, so we just wanted to summarize by taking some of the observations that we've made from working with microplastics and organic residuals over the past couple of years and talk about possible policy and management relevance. So we're going to go through each one. The first observation is that wherever people have looked for it, they've typically found plastic contamination in organic residuals. This goes for our own study and also for the studies that we reviewed from across the world. And so I think the policy relevance here is that no organics management strategy is inherently free of plastic contamination risk if you're dealing with food waste. And if that's the case, then whatever programs we come up with to try and minimize plastic contamination should probably be applied broadly to all organics recycling and management fields. The next one is we see that there's often high variance in the plastic content measured within a single material. And this should be kind of intuitive because if you have a really clean compost pile and just one bottle cap in there, and then you go and take a sample and you happen to grab the bottle cap, it's going to look like that material has a really high plastic content, even if it doesn't. And so the implication there is that, I don't put a lot of stock in one single measurement of a material. I think if you want to get a good understanding of the plastic content of a material, you have to measure it a lot of times over a longer time scale. So if we're thinking if there's talk of, some sort of monitoring program, it's going to have to be frequent and, you're going to have repeated testing in order for it to be a fair assessment of that material. The next one is that plastic contamination rates vary depending on the food waste stream. So we saw a really high range in plastic contamination rate of source separated organic food waste in the literature from like a fraction of a percent up to 6%. And so I think that, knowing that, it seems like it's possible that some food waste streams are going to be more suitable for certain organics recycling strategies than others. Should we do one thing with a really clean stream and do a different thing with a dirty stream? The next one is, and this is a big one, differences in methods make it difficult to compare results across studies. So this was really challenging for us because since there are no standard methods, everyone uses a slightly different method, and there's some commonalities among most of them. But one of the hardest things is that folks will often use different lower limits of detection depending on what instrumentation they have available to them. So we used 0.5 millimeters as our lower limit of detection, but others have used, a 1/10 of a millimeter, 100th, 2 millimeters, 5 millimeters. And so if you're comparing values that are measuring, different amounts of plastic in the material, you kind of got to, you have to have that caveat in mind that they're measuring different things, and it's a little bit of an apples and oranges comparison. And so the policy implication there is that it's going to be really important for someone to develop standard methods for measuring microplastics in organic residuals. And those standard methods should include standard units and then also lower limits of detection. The next one is that smaller particles, in the case of our study, ones that are 0.5 millimeter to 1 millimeter in size, tend to be more abundant and more common, but the larger particles tend to weigh more. And because they weigh more, they make a disproportionate contribution to the mass content of the material. And so there was some talk last year about regulating plastic content in organic residuals in Vermont, and a lower detection limit of 1 millimeter was proposed. And this is also, this is the lowest detection limit present in any regulation of plastic content in organic residuals across the world that I'm aware of. And so knowing what we know, if the lower limit was 1 millimeter, many microplastics would be missed. Many microplastics are smaller than that. But given that larger microplastics make a disproportionate contribution to the mass content of the material, this might actually be a pretty practical lower limit in the interim if we just want to get a coarse idea of how much plastic is in a material. The next one is about shapes. So film and fiber particles tend to be the most abundant shapes in food wastes, but fragments, like larger particles, tend to contribute disproportionately to the total mass of plastic in the material when present. So it's possible that we might need to have separate, more stringent standards specific to film plastics if the goal is to limit visible plastic contamination, because film plastics can be really, really abundant and numerous and visible, but they're very light. So if the limit is a mass-based limit, you could still end up with a lot of visible plastics in your material. And then finally, we found both conventional and compostable plastics in digestate and in food waste. So we didn't detect any to a high level of confidence in our compost, which seems to indicate that they're breaking down as they're meant to under a composting situation, but not under anaerobic conditions, because they're not designed to break down under anaerobic conditions. So that kind of raises the question of, do our compostable plastics also need to be able to be digested under anaerobic conditions, or do we need to have different management guidelines depending on what type of plastic's available? It highlights the need for more work around our biodegradable foodware alternatives. Great. All right, Sarah, do you want to take us home?
Yeah, thanks for that, Kate. I'm just going to summarize some key takeaways. I think we've presented you all with a lot of information. So these are kind of some more high-level conclusions. And the first of those is that our results indicate that, Vermont compost and digestates are pretty comparable to compost and digestates that have been measured for plastics in previous studies. So there's not much of an alarm there. And then generally, composts and digestates with higher food waste, those source-separated organics, had a higher plastic count and mass than those with lower percentages or no added food waste. Also, we found a diversity of plastic types, sizes, shapes, and colors. So as far as maybe tracking the source of these plastics, it seems to be that there are quite a number of sources. And then future work that's needed, as Kate mentioned, a big one is need for standard methods and reporting that can really advance our science in terms of understanding the implications of this plastic and comparing more sites of plastic measurements and then reporting in consistent and comparable units. And then we really need collaboration with finding best methods to look upstream and prevent this plastic contamination in the first place. As compost practitioners, we often have to deal with this problem, but maybe government regulations or outreach and education efforts could especially reduce this issue before it becomes a larger one upstream. And then lastly, something that our team is doing, and Kate's really pioneering this, is doing a life cycle assessment for organics residuals to determine the best management scenarios for food waste streams. So thank you all for sitting through our presentation. Kate and I really were excited for this opportunity to share our work. And we also are really grateful to the support of statewide composters and digesters for their participation in our studies. And CAV, of course, the Composting Association of Vermont, helped out in many ways, Natasha and team, the MERGE and NSAID lab groups at the University of Vermont, and then as well as funding from the Heiser Fund, the Guns Institute for the Environment, and a gift to the university by Casella Waste Systems for more research on organics. And here's our contact information, and we're ready for any questions or comments you all might have. I think Natasha might be guiding that.
Yeah,
for us.
Happy to. Thank you both so much. I really appreciate the nuance that you guys brought in and the points you raised in terms of analysis and comparison. I think that's often glossed over and people want to jump to the conclusion, and it's tricky, right? We need to really think about what it is what conclusions we're drawing and when we're making these comparisons. Okay, so Josh asks, did you say EPS foam was a big contaminant? Where is that from, do you think? Is that from food packaging?
I can't say for sure, but I think that's a likely source of it. The ones I was finding are like the little styrofoam balls that often look like they're like dented on multiple sides, so like they look like they were once a part of like a conglomerate of styrofoam packs. And like Sarah said, they're not super common compared to like thin film plastics, but because of their dimensions and they're larger, they actually weigh a lot compared to like fibers and film plastics.
Interesting. Getting a number of questions coming in now. So one is, of course, thanks for sharing your research, but also curiosity. And I know this is perhaps a little outside of what you both were looking at, but any thoughts about the cross concerns with PFAS that might come with plastic contamination?
Yeah, I mean, PFAS is a big area of concern, I think, in like many, many areas right now. I definitely have heard that some food packaging, and I feel like in particular fast food packaging, can be coated in PFAS sometimes. We did not measure PFAS for this study. I'm concerned about that. I'm more concerned that we're eating out of it in the first place, sure, that it ending up in compost. I think that's a big issue. If it is in food packaging, I think it should not be in food packaging anymore.
Yeah, thanks. And I'll just add that there is a study that's just getting underway by DEC, Vermont Department of Environmental Conservation, looking at some of the PFAS stuff and doing some of that measurement. So I know maybe by VORS 2024, we'll have some updated data to share with everybody, or sooner perhaps. So another question is, did you notice any studies that included Nile red dye as a method for identifying microplastics? So that's a very specific question. Go ahead.
Yeah, thanks for that, Ivy. We did, there were quite a few studies that used Nile dyeing as a way to identify plastics and organics matrix. But really, it's most commonly used for aquatic microplastics. And we ultimately didn't choose this as a method because there's kind of a bias in which colors that it detects. And we didn't want to be limited by that.
Interesting. And just getting back to the trickiness of methodology, right?
Yeah, and I also might.
Yep. Go ahead.
Go ahead. It also complicates being able to positively identify the plastic type. If the plastic is dyed with this dye, the FTIR, the wavelength might come back as a bit contaminated, and it might impact the purity and decrease our ability to tell what type of plastic it is.
So this, I think you covered this in the data, but maybe you can just summarize again in terms of the plastic and organics that have been run through the depackagers versus the in composts, I guess. Yeah.
Yeah, I mean, overall, we find less plastic by weight in compost and digestate than in like raw food waste samples, both source separated, depackaged. And I think that gets to the dilution aspect that we talked about. So by the time we're measuring it in compost and digestate, there have been it's been mixed with other things. Particles have been broken apart. So in general, yeah, we found less plastic in compost and digestate than in the depackaged food waste before it had undergone any processing.
Thank you. So for the SSO results, so that's the source separated organics results, did your research distinguish between programs that accept a wider range of materials, such as compostable ware, versus ones that emphasize only food scraps? And I'm just going to tag on a question to that in terms of also if you could add a comment to anything that you noticed in terms of the type of source separation program or education that, and I don't know if you have that data, but those are things.
Yeah. So we did not measure plastic in source separated organics for this study. The values that we presented in the table were from our literature search.
And from the compost samples, though, those were theoretically composted, but from source separated streams.
Yes, in part from source separated organics. So we didn't measure that directly in our study, but if you go look back at that table and read up on some of the studies that we referenced there, you can find the studies that actually measured plastics in the source separated organics. Speaking kind of generally from what I've seen, people have detected a really wide range of plastic content in source separated organics, and there's a lot of different factors that seem to play into the plastic contamination rate. And I have seen some evidence that like programs that are more hands-on and a little bit more stricter sometimes have lower plastic levels in those source separated organic streams. It's usually not zero though. There's usually some plastics there.
Great. And Brenda, feel free to add a follow-up question if that didn't cover everything that you were looking for.
Yeah, and Brenda, that was a really good question. It would be really cool to do kind of a more dedicated study to looking at methods for source separation, or maybe education and outreach, or different ways to minimize plastic upstream, and maybe if there's a correlation for your results in terms of plastic contamination with different scenarios, that would be really cool. But it wasn't something we had time to work on, unfortunately.
Sure. Deb asks, did you compare multiple categories of food waste? You mentioned source separation, but was there a non-source separated?
No, so the reason we specify source separated with the food waste stream is to just clarify that that is a food waste stream that has theoretically been source separated. It should not have plastics in it. So we didn't look at any food waste stream that wasn't already theoretically source separated. Besides the pre-consumer ones that were still in packaging.
And Brenda just said that she agreed more research is definitely needed on how source separation programs can be rolled out to cut plastic contamination. So that's sort of what I think what we were getting at, right? Is like, wouldn't it be great to understand if any correlation there is, like what is the payback for the effort, right? In education around source separation from the beginning, right? On the front side.
Absolutely.
Let's see. So are PLU stickers considered microplastics? And if yes, what category would they fit into? And did you see them?
Yeah, so PLU stickers are often in a size range that we'd consider microplastics or mesoplastics. And those are kind of your film plastics. So certainly, and we did find some PLU stickers in this study that were counted.
Thank you. Josh Kelly adds, Vermont state law currently prohibits the sale and use of certain EPS foam, foodware, beverage packaging, meat trays being exempt, however. Also this summer, Vermont state law under the Vermont Department of Health will ban intentionally added PFAS to food packaging. So that just circles back, I guess, to Kate's comment about like, what are we eating from? Which is definitely a concern, I think, for many. And as we learn more, I think that is one thing that you guys highlighted really nicely is that this is like, yes, it's always been there and maybe we know it or we see it or some of it feels intuitive, but this is really cutting edge research that you guys are doing. And there's so much we don't know. And it really is incumbent. I appreciate your sort of connecting it to different policy approaches, because as we learn more, it becomes even more incumbent upon us to make different choices about how we're regulating, managing, thinking about these materials. Megan asks, do you think that a proliferation of depackagers across the US would hamper efforts to reduce plastic generation upstream? Can depackaging be a useful tool and not just a way for large generators to single stream their food waste?
Yeah, that's a great question. I can only speak to the waste stream that we investigated in this study. But I mean, I don't know how standard it is to mechanically depackage source separated organics, because they're already source separated, so they could theoretically go straight to a composting facility. Some of the plastic that's there would be removed by screens. I don't know if more or less plastic is removed by screening or by the mechanical depackager. It is removing some plastic that's there, so it's taking a contaminated food waste stream and it's making it less contaminated. I don't know how much less, but I think that I would be concerned if it was used as to just like single stream everything. I don't think that that's a good idea if we have really clean food waste streams. And some are way cleaner than other, like industrial food waste streams that have never even been packaged in the first place. Those are going to be really clean. There's no reason I see to mingle that with plastic. Same goes, like anytime you're able to get a clean food waste stream, I think that's great and shouldn't go backwards from that.
And there's actually interest in that point in policy that is going into effect of not commingling your more contaminated streams, your less contaminated streams. But there are also kind of examples where depackagers might be helpful for example, like vacuum sealed foods or things that are especially challenging to depackage and produced in really large quantities, maybe by grocery stores. and also kind of exemptions for things that can't really be depackaged even by depackagers, like your little like ketchup packets and things might still be contaminants, and depackagers aren't really a solution for that.
Yeah, and I'll just, we have a couple more questions, but I'll just add on for folks who are interested in more of Vermont's approach to this, just refer back to that depackager stakeholder group that was mentioned at the beginning, and that has been driving new policy around management of packaged food and unpackaged food, and it will lead to rulemaking, which I anticipate we'll get into the commingling question and when and where and how and all of that. So another question that Toby says might be slightly tangential, but curious if there have been microplastic research focused on agricultural soils where plastic mulches have been utilized. So this is, again, just maybe drawing on your literature reviews.
Yeah, there's actually quite a large body of literature looking at plastic mulching and other plastic contamination, not only in agricultural soils, also in cities, urban environments, industrial sites. Yeah, definitely much has been done there, for sure, in the literature. That wasn't kind of the focus of our studies, but yeah.
And I just wanted to circle back to the depackager question real quick. So Sarah mentioned that our group's gonna be doing a life cycle assessment next. So we're gonna try and take this primary data we've collected and use it to build a bigger model to try and understand how plastics are moving through the food system. And so one part of that is to try and understand how efficient all these different separation steps are at taking plastic out of the organic stream. And so when I'm thinking about trying to model it, I have my starting organic stream. It's got some percentage of contamination that we can vary depending on what assumptions we make. And then it's going to pass through a series of different steps, each of which is going to remove some percentage of the plastic. So this could be hand picking, it could be mechanical depackaging, it could be pre-screening at a composting facility, it could be post-screening. Each of these are going to have some sort of separation efficiency. But as long as there is plastic, some sort of plastic, some amount of plastic in the starting material, it's unlikely we're ever going to get that final material down to zero plastic content. So it kind of gets back to the importance of thinking about what we're putting in our organics waste streams in the first place, because all these like post-processing steps, they can help a lot, but I think once there's plastic in a waste stream, I don't think it's possible to entirely remove it right now with the technology that we have.
Yeah, I definitely agree. And just a quick note for folks who may not be following chat, Ben Gothier from Vermont DEC just put a link to that depackager stakeholder group page in chat, and that has a report that had a bunch of recommendations that came out of that process, as well as links to some draft policy that they're currently working through all those comments. So that's a really important piece of this conversation as well. I also want to let folks know, if you're interested in source separation programs, last year at VORS 2022, we actually did a back-to-back 2 sessions on source separation, looking at sort of like the social marketing, kind of like what are the barriers for doing a better job of source separation? What are some approaches to change, to actually meaningfully shift the culture upstream about what is getting into the organic residuals that we're trying to manage. And so those are both available. The recordings and some related resources are both available on the CAV website under VORS 2022. And just for anyone who might be interested there. And I think this has been super interesting in terms of just the differences. And I think there's so much more to do in terms of the efficacy, the real life efficacy of depackagers. So just to get back to that for a second, in terms of the different types of streams that they're managing, so is it a post-consumer source separated, but contaminated source separated? Is it single stream like you guys looked at for the the ice cream pints? Is it, like what from my research and conversations that I've had, there's a lot of variability, even down to the operator and the type of equipment that is used, right? You started off by saying there's a whole bunch of different types of depackaging equipment out there. Are you buying top shelf with all the bells and whistles and extra layers, or is it kind of like just entry level? And I think that there has been some research that shows that those differences, those specific aspects make a difference, but I think that we could use a lot more research on those particular things as well. So thank you, both of you, so much again for sharing your research and for doing this research. Grab all your friends and pull them into this research, because so much more is needed. And just once again, thank you so much, Kate and Sarah, for sharing your research, and to all the participants for joining us today and throughout the VORS program.
Thank you so much for having us.
Yeah, thank you.